Okay, everyone can hear me? Great. Thank you all for coming, and I'm excited to talk about some of our work using optical techniques. Pretty much all the projects in our lab utilize biomedical optics, whether it's in animal studies or in some of the human work that we've done. And I'm going to talk today about focused ultrasound neuromodulation. I'm glad that my talk is preceded by other talks interested in clinical and therapeutic applications, as well as in Dr. Ross's talk in functional optical imaging. I'm going to take this fourth dimension of time and run with it. So focused ultrasound neuromodulation is an emerging neuromodulation technology that has spatial resolution advantages over transcranial magnetic stimulation. This is a modeling of the field. Over transcranial direct current stimulation, which has very poor resolution. In contrast, focused ultrasound, because of the acoustics of the tissue, you can get extremely highly resolved stimulation of or perturbation of neural activity. And possibly vascular, as we'll talk about. In fact, definitely vascular. Ultrasound has been used besides neuromodulation. Its mainstay has been at higher intensities for high-intensity focused ultrasound to ablate tumors and such, and also more recently for blood-brain barrier opening and possibilities in drug delivery. Some open questions, however, are, how does focused ultrasound impact neural function in vivo? And what are some underlying mechanisms? For me, it's all about the people. So I'm going to first show some results from work done by Jacob here, who was an MD Ph.D. student and recently defended his thesis. Congrats to him. And so he was asking the question of, what does focused ultrasound do to the brain, do to brain function? And so he did a combination. He took a optical approach that used a combination of wide field fluorescence microscopy looking at in vivo calcium dynamics, as well as inline a focused ultrasound transducer that's a phased array that was able to kind of scan and choose where we wanted to zap. Questions are, where do you want to zap and what's your application there? So the system that Jacob was looking at is auditory system, the representation of tones in the cortex. And so we were looking at mice, made craniotomies, and this map here is showing the different regions where different pure tones are represented in the brain. And so a question is, when you target ultrasound to a particular representation of a tone, how does that change representation? A particular condition that we're interested in relevant to this is tinnitus, the perception of a persistent high pitch. And so can one modify spatial representation of that tone? So I'm glad Dr. Ross talked about functional imaging. Here's a background fluorescence image. This crazy GIF animation here is showing basically a movie of calcium activity using GCamp as the reporter during auditory stimulus. And the question is, how does ultrasound affect that? And as you can see, it's a super noisy image. These splotches, if you take a recording, don't necessarily make so much sense. Here's a montage of those images there. And so I'm glad that one of the previous talks was Dr. Elamari, who talked about introduced principal component analysis, while in his talk, it was applying it to RNA-seq and high dimensional analysis of biological results. We actually applied this to image processing. So there's lots going on in the brain, right? That's kind of its job there. But you can use an approach, principal component analysis, to actually look at the whole movie, not just look at an individual trace, but to look at a whole movie and look at all mice and to say, what are the major voices in it? And so the top principal components will show you basically the top spatial modes. It's not exactly that. And so rather than looking at changes in intensity necessarily, we can look at how different patterns, different modes will change. The blue dots represent pre-focused ultrasound trials. So now getting into the mechanisms, there's been work looking at the biomechanics, possibly of ultrasound. It's not as direct as electrical stimulation. There are mechanosensitive ion channels that are sensitive to ultrasound, And there's also been work looking at the vascular basis, because after all, it is the vasculature that feeds the neurons and enables them to sustain their activity there. And so I'm going to talk to you about a project that Yolanda Shen, a PhD student in my lab, has been leading, and to actually see at the vascular level what has, whether focused ultrasound has modified anything. She's using optical coherence tomography angiography, and it's able to make very high resolution images at the capillary scale of the cortical vasculature. This is a movie showing optical sectioning down through the cerebral cortex of a mouse, and you can actually see microvessels. This is a color-coded image of depth there. And using image processing and segmentation, she's able to, and this is a background showing, this is a thresholded image showing microvasculature, she's able to focus, to follow individual capillaries as the brain is exposed to focused ultrasound, and to measure their diameter and look for dilation there. So what did she find? So this cool image is a rotating rendering of the microvasculature, aided by convolutional neural networks to remove some of the axial artifacts that are inherent to OCT. What Yolanda found was that in larger vessels, greater than 20 microns, the blue bars represent the blue bars and red bars represent two different intensities, low and high. What she found is that there was not very much change in the diameter of the large vessels, or in diameters, kind of a medium diameter, 50 to 20, but in the capillary range, there was a significant dilation in response to focused ultrasound. Apparently restricted to these small vessels. And because we're able to look in depth, we're also, these violin plots are looking at, are basically summarizing the diameter distributions of all the vessels as a function of depth in all the animals we are looking at. And so you can kind of see that the bottom weighting here on the left, on the y-axis, you have diameter. The smallest diameters, in the green, you have the pre-ultrasound, and in the magenta, the post-ultrasound. The smallest diameter capillaries vessels are the ones that experience the largest change. And we're interested in this, and this actually is kind of going to guide our next steps in terms of mechanistic exploration, because what are the small branches? So they are high branch order vessels. So in this cartoon on the right side here, you have a penetrating arteriole. Every time there's a branch, we count that as an increase in the branch order. The capillaries are high branch order because they've already been branched many times. And so if the capillaries, though, are the ones that are changing in their diameter, how is that happening? They don't have smooth muscle cells. What do the capillaries have? They have parasites, which are motile, and are therefore are they involved in, is it modulation of the parasites that could be involved in the selective dilation of those? And so just the last slide here, ways we're going to be looking at this is actually in vivo, looking at following the patterns, following the distribution of neurotrace, which is a stain that apparently, so you can use it in vivo, and apparently it's highly selective to parasites. It was found recently. I wish it were a little darker in here, because I spent a lot of time trying to bump up the contrast in these. But what you can see is the scale bar there. This is a image in vivo, wide field. The red, those are blood vessels, and you can gauge by the scale bar there whether you're looking at precapillary arterioles or smaller. And the green is the neurotrace labeled parasites. And so you can see on the small vessels these kind of elongated parasites there that you get at the precapillary arterioles. And so we're going to be using this approach, combining OCT as well as monitoring parasite morphology to see the level and the nature of involvement of of those cells in that. And so I want to end by acknowledging the people who worked on this, Yolanda Shen, Jacob Hahir, as well as our collaborators on the engineering side at Washington U, St. Louis. Particularly, it's the lab of Chao Zhou. And I will end there. Thank you very much. Trying to make up time.
Great, we have time for some questions. Any questions from the audience? While you're thinking about it, I'll ask you a question. We already have a question. Great. Oh no, I was just going to ask you, it looked to me like in the estimates from Yolanda's work of the dilation produced by different power ultrasounds, there was really no effect. I mean, a tenfold increase in power didn't change the dilation.
Yeah, absolutely. So the blue and the red, those correspond to, as Dr. Leonard says, an order of magnitude difference in the ultrasound intensity, 1 watt per centimeter square, 10 watts per centimeter square. I should say this is basically below the threshold for actually feeling this. You know, we're not talking HIFU or ablating anything. But so a question is like, what is the dose dependence like? And indeed, there is a small significant difference at the 1 minute point after focused ultrasound, but it's really not that significant. And this is great to know because you obviously, you know, for therapeutics, want to have the minimum exposure that you can. And so, yeah, the question is, have we just kind of saturated whatever? Have we getting to this? Have we, you know, with our dose of ultrasound, just saturated whatever mechanistic underpinning there is? And that is what I would assume, but we can maybe investigate that some more.
Very interesting. Yes.
So I have, it was a very nice talk. I have one technical question and then a kind of a biological question, just to, you know, it's a speculation based on what I know of vessels in cancer. So the technical question is, what is the depth of penetration for the focused ultrasound? Do you lose a lot of resolution as you start to go deeper into the tissue? And then the biological question is it possible that it's not necessarily an effect on the parasites, but maybe the ultrasound is damaging the extracellular matrix that surrounds the vessels, and with that damage, it may be causing a relaxation, and simply the pressure is causing a larger diameter?
Yeah, no, great question. So I guess first off, in terms of the depth, let me go to. I'll first go to here. So one of the nice things about the acoustic mode of stimulation is that you can, depending on the wavelength, you can target centimeters within the brain. So the trade-off, however, is that as you try to get deeper penetration, what you do is use lower frequencies of ultrasound. The challenge, though, is that the lower the frequency of ultrasound, the fatter the focus. Am I like right in your ear there? Oh, sorry. Sorry about that. Should have roped up that seat there. So yeah, so the lower frequency that actually kind of expands it. But you can at the frequencies that we're using, you could get the central focus to something like millimeters. This obviously makes small animal work, you know, the interpretation therein, you know, a little iffy because you're stimulating a lot on the brain. It's more like a spear. But certainly axially it's much tighter there. I want to be mindful of the time. So the next question was about possibly disrupting the extracellular matrix there. Yeah. So one of I mean, exactly on that point, ultrasound has been used for permeabilizing the blood-brain barrier for possible delivery of drugs. And the way that this in order to actually get that kind of perturbation of the blood-brain barrier, at least at a large scale or intensely, is by using microbubbles injected intravenously to kind of, as like a kinetic contrast, to kinetically amplify the acoustic perturbation. We have monitored for BBB leakage by using Evans blue, which is it's a dye that you can inject. Apparently, you can also inject it intraperitonially and have it used that way. But your animal gets, and the student gets, very blue. And we look for leakage and permeability that way. And we don't see that. It is possible, however, that we're getting a little bit of that we have not detected. We certainly could look with more sensitive, higher resolution approaches. For example, with 2-photon microscopy, that's probably a better way to do that. And was there a third part of your question, or that's that? Okay. Thank you.
Dr. Fisher, I have a question. This is Andrew Mayer from MRI.
Hi.
Hey.
Hi, how are you?
Good.
Wondering, speaking to that last that you're making, have you guys thought about looking at all at transcellular versus paracellular processes as an additional way to look at the blood-brain barrier opening and leakage with something like electron microscopy? Do you guys have the capability of doing that there?
Oh, I mean, that would be great if we could get electron microscopy level resolution to look at the mechanisms. I think it would be great if we use, for example, super resolution, maybe simulated emission depletion microscopy, and certainly I take the cue from Dr. Entenberg about possible super resolution approaches. Yeah, if we could do that in vivo, that would be amazing. One of the challenges of functional measurements, certainly with neuromodulation, is that you need to make these measurements in vivo, and the time, the time course of 10 minutes will make all the difference. So if we could do that in vivo, I wish that we could have electron microscopy level measurements in vivo, but we'll take as sharp as we can get. And it's super important, as you said, because the mechanisms are, because we can't see a serious BBB permeabilization, that doesn't mean that it's not actually happening, and it is a mechanical phenomenon. Well, a part of it is likely a mechanical phenomenon. We'd like to be able to see that. So, thank you.